Hello everyone. Today we are going to learn how to identify the homologous and analogous organs in animals. Homology is an evolutionary concept where a given organ in two animals shows the same evolutionary origin but not necessarily the same function. Homologous organs are those similar in structure and position or function and are derived from a common ancestral organism. Let us take a specific example, the forelimb of a human and a bat. Anatomically, a typical vertebrate forelimb is made up of 5 segments. The upper arm represented by the humerus, the forearm represented by the radius and the ulna, the wrist represented by tiny bones called as the carpals, the palm, it is represented by metacarpals, and then comes the digits which are represented by what are called as phalanges. So, if you look at the skeleton of the forelimb of human, the upper arm is represented by humerus, the forearm is represented by radius and ulna, the wrist is represented by tiny bones called carpals, the palm is represented by slender long bones called as metacarpals, and the digits are represented by phalanges. Now, look at the skeleton of the forelimb of bat. Here also you can see the same segments, that is, the upper arm is represented by humerus, the forearm is represented by radius and ulna, the wrist is carpals, the palm is represented by metacarpals, and the digits are represented by phalanges. In both the skeletons of the forelimbs of man and bat, all these 5 segments are recognizable. Their positions are the same. The anatomical structure of these two organs follows the basic pattern, but there are specific differences between the two. Let us look at the differences now. Human humerus is strong and stout because it is meant for load bearing, whereas bat's humerus is thin and long because it is meant for reduction in weight and providing extended support to the patagium. In the humans, radius and ulna are long and strong, so they can overlap to make supine and prone movements which are ideal for tool using, whereas in the case of bat, only the radius is present as a thin long bone. The ulna appears just as a splint. This modification is for the reduction in the body weight so that the bat can fly easily. In the humans, there are 8 round carpals which can glide over one another to allow twisting movement of the hand. This again helps in tool using, but in the case of bats, there are only 2 carpals. Less number of bones reduces the body weight which enables the bat to fly. In the humans, the palm has 5 long metacarpals which support a broad flat surface. In the bats, there are 5 very slender and elongated metacarpals to support the patagium. In the human, the digits have 14 phalanges, 2, 3, 3, 3 and 3. These phalanges can fold inward, enabling grip, whereas in the bat there are only 9 phalanges, 1, 2, 2, 2 and 2 is the phalangeal formula in bats. These are thin and long to provide support to the patagium. Even though the forelimbs of bat and human are similar with respect to origin and basic structure, they exhibit these differences because they got modified or evolved for different requirements in different habitats. So, the human forelimb is primarily evolved for tool using, whereas the bat's forelimb is evolved for flight. Therefore, homologous organs shows divergence in evolution. Now let us learn about analogous organs. Analogy is another concept where a given pair of organs of two animals show similarity in function but not in evolutionary origin and structure. Let us take an example, the wing of an insect and the wing of a bird. Let us look at the differences of these two organs. An insect wing originates purely from ectoderm, but a bird's wing originates from ectoderm and mesoderm. An insect wing is just an extended part of its exoskeleton without any subdivisions, but a bird's wing has 5 recognizable regions in the skeleton. A wing of an insect is covered by microscopic scales, whereas the wing of the bird is covered by feathers. The muscles that operate the wing of the insect are within its body, whereas the wing of bird is operated by muscles within the body and within the wing itself. Even though these two organs are different with respect to their origin and structure, both these organs have evolved for the same function, that is flight. So, these two organs are analogous because anatomically and structurally they are different from each other, but they are similar only in function. Let us look at another example for analogous organs, the mandible of cockroach and the lower jaw of rabbit. Let us see how these organs are different from each other. Mandible of cockroach is made up of chitin, but the lower jaw of rabbit is made up of bone. Mandible of cockroach originates from ectoderm and it is a modified leg, whereas the lower jaw of rabbit originates from mesoderm and it is a modified gill arch. Mandible of cockroach is well sclerotized and the molar area is specialized for grinding, whereas the lower jaw of rabbit is thin and flat and well ossified, and the teeth are of different types, incisors for cutting, premolars and molars for grinding. But the function of these two organs are the same. Both are meant to grasp, crush or cut food and to defend against predators or rivals. Both the structures are analogous to each other as they perform a similar function but are structurally different. Therefore, analogous organs shows convergence in evolution. In conclusion, a pair of homologous organs resembles each other in their structure and origin, but may or may not resemble in function, whereas a pair of analogous organs resemble each other only in function, never in structure and origin. Thank you.